Hello.
Hi, Bradley. Just a minute.
Okay. Hello.
Shall I start with the update?
Yeah, go ahead.
This week I have trained the SAM model. It's giving best accuracy, I think so. I'm a little bit happy with that.
Good. So that's your Kaggle account. Oh, you have a data set there you're working with.
Can you see my screen?
Yes. Yes, I can.
Oh. Yeah. Here you can see, Bradley, it is giving 0.99.
Okay.
I've trained it up to 10 epochs only. Whatever the loss function I am using, it is dice loss. So dice loss generally lies the loss value between 0 and 1. If it gives near to 0, it's the worst model. I mean, it's not predicting the correctly. But here we can see it is giving 0.99, which is approximately near to 1. The model accuracy was good and it is predicting perfectly. Maybe in next week meeting I will add the visualization of all these model, train model. Right now, at starting while I was training, I got some memory issues and GPU issues, but I cleared them out and I have trained it up to 10 epochs. Batch size was 4 and model was able to learn. This can, I mean, yeah, model was completely learned here.
Okay.
Here we can see all the data, how much data it has used, etc. But the final thing is, it took around 3 days to train it on batches. So yeah, and I was working on the paper. I was reading some papers. I was writing paper mainly for C. elegans based segmentation, because C. elegans, there are no research papers on C. elegans regarding the cell segmentation part. So I was reading some past research papers about C. elegans when they are in between when they are going through embryogenesis.
Right.
And in that domain, we are using deep learning to segment these cells. That's the whole paper about. I'm creating like comparisons. First, I need to understand like why it is hard to segment C. elegans and all these things. I have some 3DMM robust model paper on C. elegans, and QCN one another paper. C Shaper was another paper which you have shared, and I'm trying to implement them. I mean, not implement them. I'm trying to understand what is the main problem of segmenting C. elegans, and those matter I will use in our paper, but in my own words, without getting caught in plagiarism and all this stuff.
Here.
And I mean, these are the results actually. I need to show it to Mayuk actually.
Okay.
But before I showed it to you, maybe by next week meeting I will show it to Mayuk and confirm it. But the model accuracy was good. I mean, the loss function was very high. I didn't expect it, like it would come around 0.99. I expected it around 0.80 or something. But it starting only, it started working fine. I don't know really whether I'm wrong or right, but I will discuss with Mayuk and I will let you know. But finally, the model was able to learn, and we need to create some visualizations for papers like pictures, graphs, etc.
Right.
That's the only.
Yeah, I think that's true. So those look good. Those results. And then the kind of thing you usually put in a paper you report. I think in this case you're going to have your loss function versus your epochs. So it goes from like 0 to 10, and then watch. If you, I don't know if you have the loss function for each epoch, but it should.
Yeah, I have the loss function for each epoch. You can see epoch 6 it has 0.9589. Just it is changing around this decimal part. It's not changing in the upper decimal part.
Yeah. That decimal. Yeah, it just basically is asymptotic to 1 or 0 or something like that. So, but you'll just you'll have the thing. It'll show sort of as it's sort of reaching its maximum learning capabilities. It'll be flat, it'll become flat. So
I think the model showed more accuracy because it was trained on so many images, and the model, this model, the SAM model was trained. It is giving zero shot performance on general images only, but its microscopy, it is more even easier to segment it. That's the 0.99 rather
yeah,
than showing a very low value. So yeah,
right.
we can find this. I was thinking to write a paper in general way and post it as a preprint in RZ.
Yeah, yeah. So yeah, that's good. I want to go over. I saw you were working on that a little bit. The paper.
Yeah,
We should probably plan out the different graphs that we want to put in. There obviously be graphs like the loss function graph. There will be other types of graphs that we'll want to make, and then we'll be able to make a report on it. We'll probably also have some other performance metrics. Sometimes people will like train a model on some trivial data set just to show what the model can do. So it's like something that's really easy to segment, like a bunch of circles that are just basically really easy. There's no background. The background is very obvious, and then all of that would be like trained, and it would be really quick. But to show like that this is actually doing something instead of like just picking up noise. Yeah.
okay. I will try to create some visualization for it, like graphs, pictures, etc. I'll try to host it like on Hugging Face. I will try it. Uh, SAM model was not supporting to host it on Hugging Face in Hugging Face spaces. Maybe I can use some NAPRI, some segment anything have provided, like Meta AI provides some other thing. I'll try to go through into it and I will try to implement that. It's a NAPRI library. I will show it to you next week meeting in a slide, maybe. Yeah.
Yeah.
Before that, I just want to mention Bradley, do you know like how to host a preprint in RZIF?
Yeah. Yeah.
I was just thinking to host it in RZIF because this work should be in sessions, like not in single go. I can't host this all work as a paper, but we need to more develop this thing. Not only for C elegans, we need to adapt it to other cells also. Not only C elegans, like mouse cells, etc. We need to train that. Due to GPU issues, I haven't trained on all of them, but I will try to train them on other cells also if I get a chance.
All right.
So we'll keep this as a phase of work, and we'll first at least host it on the RZIF as a preprint or research.
Yeah.
Then we can shift it to other things.
Yeah, that's fine. Yeah.
But I think when I thought of creating an account in RZIF and I created, but it was showing like some registered author should invite us to post our work. I read something like that. If you don't mind, can you go through that? Let me know.
Oh yeah, yeah, I can do that. That's okay. Yeah, so I think that's.
Sorry, go ahead.
Oh, so that's pretty much what you've been working on. It looks pretty impressive. Why can't you host it on Hugging Face?
Hugging Face spaces. The model was around 375 million parameters.
Oh.
It's not a small model, and Hugging Face spaces provides only up to some extent level to host it. Not like it's a big model, right? On free version we can't host it, and model will not also work fine as right now we have 3 models over there which are working fine because the parameters are very small. But whatever the SAM model it is there, it has so many parameters, and the free version type will not satisfy that.
Okay, yeah, that might be a problem where we might be able to host it. I have to think about that a little bit.
Okay, but we can do this thing. We can run it on local machine and see like it works or not.
Okay.
If any to see the output of the model or etc., they can just take the model, run some commands in their terminal, and they can see it by themselves. But host on Hugging Face would be a problematic thing.
Yeah, I understand.
I will try to show the demo of that, maybe.
Okay.
At least you will understand next week.
Yeah.
those are my updates for this week.
Okay.
And yeah, I'll try to host it on at least local machine, and I will show it to you whether it's possible or not.
Okay. Yeah, that sounds good. Yeah, a lot of the large language models are sort of having that same problem where you want to run them on your local machine, but you want to be able to like also host them in different places. And they're pretty big models, but like, working with them is a little challenging, but still, but that sounds good. I mean, that'll be good to have, in place. I think this is a pretty big improvement from what we've had before. So, and again, we'll have to plan out the figures and the tables and things. There are certain things we'll want to report. So, when I know you've probably read a lot of papers, but about, in that area, but to look at some of the papers and how people report things. So if they're like, different ways that different graphs that they have that you need to have to report. Like I said, you have the epochs against the loss. Yeah. You want to have like these control conditions, like very simple things, or sometimes people will use Gaussian noise to like, just kind of say, this shouldn't segment, or this should segment very easily. And if it does or it doesn't, then you know that like it's not picking up on it's picking up on the right kinds of things. So you'll see the loss function kind of saturate over time, but it's actually learning instead of just kind of like improving. And there'll be other things too, like some statistics that we'll want to get, but I don't know what the typical tables are that people make. We'll just have to figure out what those are. So go through like some of the conference papers that they have for like machine learning or deep learning, and look at like what people, especially like I don't know what the SAM model paper, what they're reporting, but that might be the best sort of starting point. Because if we're doing like something that's related to the original SAM model, I want to give the things that they reported, at least as a point of reference for people.
Okay, Bradley. I will definitely make some visualization for graphs and pictures maybe by next week, and I will show it to you. Maybe I'll try to use WNB library to get these all visualizations, and I will let you know.
Okay. That sounds good. Thanks. All right. Thanks for the update. Hello. We have Susan and Dick and Lukas joined. So, hello.
Hi. I think I'm IA this morning as I'm having trouble. I don't know if I'm having trouble or not. It just turned out.
You're coming through clearly. Yeah, you're coming through clearly here.
Yeah.
I'm coming through okay?
Yeah.
Well, I'll shut everything off, but I'm IA this morning.
Okay. All right, great. Thanks for the update. Yeah. How are you, Dick?
Okay.
Yeah. So I've been working on the paper. I'm almost ready to send it out to you guys. So it'll happen this probably today, later on today.
Oh, good.
Yeah. And Lukas finished up with his analysis. So thank you, Lukas. And then I've been working on some analysis and that. Lukas, is there anything you wanted to say?
No. And I actually wanted to say, so you're gonna send the paper to me as well to check it, like for review, because I haven't seen. Like I've sent you the details stuff, but I haven't seen the updated version of it.
Oh no, that's coming today sometime probably.
Okay.
So just be on the lookout. Yeah. So I think Lukas did a pretty good job. You're still like an undergrad, right? Or you're an undergrad?
Yes.
Okay, yeah. So you did a pretty good job on that, getting a lot of the data together and going through it's not an easy thing to do. So it was impressive. So that's good. All right. Yeah. Sushmanth was talking about documentation and the importance of documentation. And I know that Mayok said that he would fail Sushmanth if he didn't do proper documentation. So, but it's very important, and it's something that we talk about a lot in open source. And I know Himanshu's not here yet, but I'll kind of use this. Hopefully, he'll come in as we're talking about this. So there are a lot of examples of documentation that you want to use, and I have some other GSOC students. We have an open source meeting on Fridays where I actually went over some of this stuff. Went over 4 of these documents, and then one of the people from our lab group, he actually gave some more links, some more examples. So we're going to go through some documentation examples for open source software. So let me share my screen here. So, there are a lot of things that go into good documentation. Obviously, you want to have be precise and be clear about what's in the repository, but you also want to have different types of documentation. And I think we talked about this earlier about, you want to tailor it to specific groups of people you want to use your software. So for example, if you have people who know exactly what they want out of it, like for example, the case where you have something you want people to run on their local machine, it might be a little hard for someone who just wants to segment some cells to run the program versus someone who wants to benchmark the software. You just have different skill levels and they want different things out of the software. So it's important to cater to those different groups of people. And you kind of have to think about who you want to get to use your software, who's most likely to use your software when you write the documentation. So, you might write one piece of documentation for people who maybe just want to use it, maybe just a down and dirty sort of segmentation, and they don't care so much about the performance. But other people might want to, they care greatly about the performance. They're basically, professional computer scientists, and so they want to know about that. So you offer documentation in both of those areas. So the first example I'm going to give is there's this article we went over in our open source meetings, The Anatomy of a Great Open Source Documentation, How to Document Your Projects on GitHub. And this kind of goes over some of the points that they talk, that are important for good documentation. With documentation, it's usually something that you write up. You maybe give examples of how to use the software. You describe what it is, what's in the package. And the idea is that your code becomes more understandable and reusable. People can understand how it works. They can use it for their own purposes. This will help with recruiting people for open source participation. So, I think with what we have with DevoLearn, enough people had seen the project and they were able to basically understand it. But there was a lot missing, of course, but you want to get it to the point where people can identify what they can contribute to. What are the missing pieces or what can be improved? And some of that involves documentation and making sure people understand how things work. You also empower people who maybe either want to understand the code base or they want to use the project, and empower them so that they'll be able to use the software. And so that's just basically instructions on how to use it or cases of how to use it or things like that. And so but documentation can also help build the reputation of the project. So there are a couple of examples of GitHub documentation that are really well done. This one is a Space Invaders game that someone made. This one actually is I guess it's a class project that someone made. So this is an example here where they just have this project navigate the Mars rover. That's the first project. Help the Mars Curiosity rover find the shortest path between 2 points. while avoiding obstacles on the way. So they have this software here that you can run, and you have these different projects that you can do, or these different like sort of demos. And so this basically shows how to do this. It gives you an animation, and so it shows you kind of how to do the task, how to use the software, what kinds of things you want to do with it. And then, so it gives a lot of graphics here. It gives a summary of the search algorithms. It gives lists some other features, and then it tells you how to run the software. And then, this is where, not everyone has the same hardware. So you might suggest that people run it in a container, or that they run it from the command line, or they run it from there are different ways that people want to start the software and run it. So these are, including these in the documentation is important. Sometimes, what you can do is, if people want to use it for different things, you might have different tabs. So you might have different markdown files where you have, this is for people who want to get into the program and work with the code. There's another piece of documentation for people who just want to run the software and get some statistics out of the images. In other words, they don't care so much about, the software details. They just want to run it. And so those are things you can do. And so then, you list the things that are in your library. You talk about like the different classes in this program. So this is an object-oriented programming paradigm. That, you have different classes that are defined in the software, and you can document those as well. Then there's this health bridge, which is a Project Health Bridge. This is bridging the gap between patients and doctors. You'll have a link to the website. You can view the demo. You can report bugs right from the documentation, or request a feature. And usually those kinds of things are like if we use the issues tab in GitHub, you can go into the issues and file an issue, and we can create in the DevoWorm or in the DevoLearn repository, we can create a template for people to create issues. So I don't think we have a bug template set up in there or a feature template, but that's something we could do pretty easily. Set that up and, have like people just fill out a simple form, and it goes right into a GitHub issue, and you can address it. And then of course you have the different things about the project. So you have like a table of contents, and then you have a lot of visuals again. You have a lot of like kind of main points, what the project does, what is it, how is it built, how do you install it, and so forth. And then there's this third project, which is Resonance, which is a content-based music recommendation service. Again, they have visiting the website, viewing the demo, reporting bugs, requesting features, a table of contents, a project description, and so on. So this is, how you build, open source documentation. I can provide these links in the Slack, so that's fine. But you just have to think about this as like an exercise, and how do you communicate to people what your software does, how to use it, and so forth. So you start with a README file, usually in the repository. So that's usually the convention, is to provide a README file, which is this file here. And you can have other documentation, but it usually starts there because that's how people expect to see it. You want to host it kind of in the same place so that you don't have to, people can find different files if they want. So that there are strategies of doing this, but basically you start with this README file. It's the project storefront. And, it'll help people navigate your repository. So then, there are these other style guides that people have made for documentation. Basically, there's this idea of minimal viable documentation, which is where you have this small set of fresh and accurate docs, which are better than a sprawling loose assembly of docs. And the thing you have to remember with documentation is, when you create documentation, someone's always going to have to update it. So as the software evolves, people have to update the documentation. So if you have 10 pieces of documentation, you'll probably have to update 10 different documents as it's updated. So you want to be able to identify your docs, and that usually means making a small set of docs, and make sure that you have a strategy for updating them. Because if you have documentation that goes out of date, it's not good. You want to, don't be afraid to delete dead documentation. I know we have documentation in there, which probably needs some trimming. We need to change things, but we maybe also need to get rid of old materials. And that's true, I think, in any project. But when we're improving upon a project, especially, the new version isn't there. Well, things in the old version is not relevant to the new version. And we'll have to do that with the preprint that exists now as well, and those sorts of things. So that's what we need to do. And then we don't want to over obsess about documentation. Just put something out that's readable, or start with something that's readable, and then we can modify and edit from there. So don't think you need to make this perfect document right off the bat. It just needs to be all the relevant information needs to be there, and then we can worry about the design of it and style of it later, or in different drafts. And then so you're telling a story about your code. You're basically you're walking people through it, what it's about, what the components are, and those sorts of things. That's documentation best practices. Then finally, there's this idea of a document management system. So a lot of the whole idea of documentation comes from this document management system approach. So when you have documentation that's more than a single README file, which is important for a lot of bigger projects, you'll want to have this sort of document management system approach. Take this document management system approach, which means you have to manage the documents along with the code. And it's important because you need to update the docs and everything. But it's also important because you want to keep track of all the docs and make sure that everything is linked to, everything is updated, and so forth. Now, GitHub takes care of a lot of this. This is just kind of a generic stub on document management systems, but you're basically working from this set of documents, and you're also integrating your code into it because you're going to have things like metadata. So if we have a data set that we analyze or use as a benchmark, we want to have data about that data set. We want to integrate the docs with the code, with the comments in the code, and with the data sets. And we want to have all these. We want to be able to store them in a place that's good. So that's all I'm going to say about documentation. I think that's I presented this to the open source meeting, and they got a taste of it, so I wanted to present it here as well, in limited form. And thank you to Ankit Grover for pointing out those repository examples. So, any questions, comments? Okay.
Is that online? Sorry, I missed.
Oh, was that?
Is that information online?
Yeah, it's on.
DevoLearn? No.
Yeah, it's usually I put it in our Slack. You're not a part of our Slack, but it's just a bunch of links from the Wikipedia and from other blogs and things like that. So, but usually, documentation is online. Oh, yeah. We have a couple comments. Oh yeah, link. Okay, I'll put the links in the Slack. There are several links that are involved in that. So, okay, I don't know if I'm on.
Something I think I should do eventually
with for your microscope projects, or
for the Tensegodi project for the programming. end of it. Yeah.
Oh yeah, yeah. Yeah, that would be a good idea actually. Yeah, it's something we can work on in the group too. If you're interested in. But yeah, usually we could even host a GitHub repository on the tensegrity stuff if you're interested. Yeah.
Okay, I just need to finish it right now. And,
Yeah.
the first part of it.
Yeah.
Then I have to figure out the equations.
Oh,
for
right.
it.
Yeah.
Yay.
Yeah, walking through equations is like another thing you can put in documentation form, and it's a little bit different than in papers, because in papers you usually put them in a very formalized style. You put them like center, and you tell everyone what the parameters are or the variables, but you don't really kind of walk people through what the equations mean. Like if you were in a course and someone were at the board and they were showing the equations, they might show the derivation, but they also might show what the components do, the different parts of the equation, and then show what it's related to. And you can do all that in a documentation setting, whereas it's not really acceptable to do that in a paper. It kind of detracts from the paper itself. So
yeah, you usually dump those in an appendix. So
right, yeah,
yeah.
All right. Okay, so that's all I had to say about that. Now I wanted to present a little bit on. So there's this. We've worked on CompuCell 3D. I remember very early in the history of DevoWorm, we were interested in using this program for modeling cells and modeling. So it's an agent-based modeling platform for cells. It's cell-centric, so you can model tissues, you can model different types of biological systems at the level of the cell. So I actually visited their headquarters a long time ago and gave a talk there. But they, it's a software that I've tried to get people to use several times in the group. And each time we've tried to use it, no one's really been able to advance anything on it. And I guess it's not very easy software to use. It's very hard. So they have these workshops that are like kind of help people use the software. And so this happened recently where they have a summer workshop and they have this discussion about different topics. So you download the software, you can use it, but it's very hard to figure out kind of how to implement your model system of interest. And so this workshop was recently done where they had all these different topics: network modeling, compartmental cells, links, cell crawling, convergent extension, tissue folding, etc. And you can implement all these in CompuCell 3D. Now Morgan, who is not here today, pointed this out to me that this was going on, and we had a discussion about it. So there's a software, CompuCell 3D. You just look it up on the web. You can find the link, and you can download the software for free. And it's a software where you can model a lot of cell sort of behaviors. So there's a lot of physics. There's a lot of different types of cell-cell interactions you can model. You can specify a lot of parameters like that. And so some of the projects they have in the software, they have different modules that you can download. One of them is I think the chick embryo, something about, I think, convergent extension and chick embryo. There are some other examples from like the liver and other things, where they're actually modeling tissues. They're modeling the cells in the tissues. And because it's like a agent-based model, it's like a cellular automata, you have these cells that interact on a grid. And in this case, it's a 3D grid. And so you're able to get a lot of, you're able to do capture a lot of things in that way. So this is something that I think people would be interested in. They also have a YouTube channel, but their YouTube, like the last videos that they had are a couple years ago. So I didn't know if that was, it's probably relevant to today because I don't think the software changes that much. but this is something that people might be interested in. Again, we've tried to do a couple things with this, but it's just really kind of hard. There's a high learning curve on it. So it's people have to be really invested in it to really kind of get something out of it. But I'd like to do something with it if people are interested. If anyone's interested in working with CompuCell 3D, if anyone has any specific questions they'd like to ask with the software, especially, I think, because you can learn the software and just play around with it, but you need to really have some reason why you're using it. And so there are a lot of questions that we have that are really interesting. I think that could be answered with the software. If you're interested in a biological question regarding like how cells interact in tissues, or how tissues interact, or how organs interact, those are kinds of things you can answer with this software. And there are a lot of different plugins and things you can do to really kind of refine your model. You can also train it with model or data from different sources, physiological data, etc. I'm not as familiar with how to do that, but that's part of the fun of setting up these kind of models. So it might be useful. We might be able to do something with that.
Yeah, it's. I was going to use Morpho for what I'm doing. My problem is I need to know the exact equations that go with this. You can't have them hidden in the background and play with it. For my purposes, I actually need to know the process, including the equations.
Yeah.
Anyways.
Yeah, it's a lot of these programs, they allow you to set up equations in different ways, or set up like interactions in different ways, and so it's they're like, this is an open source platform, so it might be easier or might be even harder actually to do that. So it's hard to say, but yeah, I like where you have these flexible modeling platforms where you can apply just about any question to it, but you have to figure out for your specific question what you want to actually ask, and then you have to figure out how you want to represent it. So it's like, I have a cell, I have cells that are interacting. What do these interactions mean? If I have a standard equation, what am I plugging into it? And that's kind of a challenge, because you have to understand your system pretty well. But yeah, so that's something we can follow up on perhaps in the future. Then I guess I wanted to talk about these papers here. So this is getting into, again, with the synthetic biology field, where people are creating different gene circuits, and they're making things with them. So they're able to take like a gene circuit that's well known, and they're able to sort of design new circuits, because they know that they have a promoter and they have some coding DNA, and they can make gene products, and they can do things like make enzymes, but they can also design these things because they know the sequence, and make new things that we've not seen before. So people have made different circuits where like it expresses like letters and shapes and things like that, which are kind of fun little toy models to work with. This is an interesting paper where they talk about regulating synthetic gene networks in 3D materials. So this is actually where they have 3D materials, and there are a lot of biological applications for medicine and for other things. So this is, I think they're working with in a cancer institute, or they're working with a cancer institute and an eye institute. So the abstract reads, combining synthetic biology and material science will enable more advanced studies of cellular regulatory processes, in addition to facilitating therapeutic applications of engineered gene networks. One approach is to couple genetic inducers into biomaterials. So this means that we have these gene circuits that we're making, and we can, they're regulated, they're self-regulating. You're inducing it with certain things, like, so you have this inducer, which is where you stimulate it and it induces a biochemical cascade, and it produces what you want. But you can do that in a dish, or you can do that sort of in an isolated environment. What they're doing here is they're integrating it into biomaterials. So they're able to generate 3D microenvironments that are capable of controlling intrinsic and extrinsic cellular events. So that means that you can control sort of the outer environment of the cell. So the cells are in this sort of extracellular area, and you can control those kind of events. But you can also control intrinsic events within the cell. So there's, you can create these circuits that will allow you to sort of shape those types of events. Here we have engineered biomaterials to present the genetic inducer IPTG with different modes of activating genetic circuits in vitro and in vivo. So they're able to activate it both sort of in a cellular model in the genome, and then outside of that. Gene circuits were activated in materials with IPTG embedded within the scaffold walls, which means that they're able to get this chemical in the scaffold. They have a scaffold in their 3D microenvironment, and they're able to express it in the scaffold or chemically linked to the matrix. In addition, systemic applications of IPTG were used to induce genetic circuits in cells encapsulated into materials and implanted in vivo. So they were actually able to induce a circuit in a cell. They were able to put the cells in a material, and they were able to build a system around that. The flexibility of modifying biomaterials with genetic inducers allowed for pattern placement of these inducers that can be used to generate distinct patterns of gene expression. So they're basically trying to embed cells in materials, and they're inducing some gene expression that allows for maybe to aid in pattern formation. So one of the things they want to use this for are different types of controlling diseases, treating cancer, promoting different types of biofilm degradation and things like that. So if you were thinking about, if you're using this for therapy, you were to implant one of these systems into a patient, you would be able to affect, you could implant it near a tumor and reduce the size of the tumor. Or you're able to implant it in some sort of gene therapy context and help that process along. So that's where they're going with this. And then what they want to do is they want to integrate it with biomaterials, because a lot of these medical contexts, they have biomaterials that need to be in place, to serve as a scaffold, or to, serve as sort of a, if you're implanting some sort of mechanical device, you want to be able to interact with that without the body rejecting it. So that's why they want to do this. The flexibility of modifying biomaterials with genetic inducers allows for the pattern placement of these inducers. Together, these genetically interactive materials can be used to characterize genetic circuits in environments that more closely mimic cells' natural 3D settings. So what they're trying to also do is they're trying to put these cells, or get them to behave in the kinds of settings that they're in the body. So usually we just kind of get these systems to work in a dish, so it's like a two-dimensional layer, and that's fine, but that doesn't really have a lot of applicability to medicine, because in the body, the cells are in this three-dimensional space. They're interacting in 3 dimensions, and they're interacting with, materials in 3 dimensions. So, your different tissues are interacting, muscle with bone, and so forth. So they want to explore complex cell matrix and cell-cell interactions to facilitate therapeutic applications of synthetic biology. So this is a nice paper where they talk about putting this into these biomaterials. So this is an example here of they're doing this in a dish. They're able to, they always do this validation where they look at the markers that they're looking for the expression of IPTG in these cells. So they put this circuit in a cell, and they see that it's able to express this marker, and that's how they can validate that the circuit works. Then they're able to do these, they use these electrospun PCL fibers, and they embed them in these fibers, and it's able to sort of survive in that kind of environment. And then this I is where they have RT-PCR expression of EGFP, which means that they're getting this upregulation of the GFP signal in these different contexts. So they have the two-dimensional context in the dish, they have it embedded in fibers, they have it embedded in sponge, and then they have it embedded in some other vehicle things. So they're actually able to show that that's more or less consistent across these cases. And then they do these experiments where they show the number of days in culture, so they want to show that it's stable at different dosages over that time. Yeah, so this is just basically a demo. I'm wondering if there's a picture of them putting it into here's where they're showing it in the context of a mouse. So this is where they're talking about the therapeutic applications. This is an example of local induction at the top with this little mouse here. This is like supposed to be inside the mouse, this little cube. PLGA sponges with and without IPTG. So this is where they put in like some sort of sponge inside the body of the mouse containing CHO cells. So they have this sponge, it's embedded with these cells. It's stably transfected with this GFP, so that means they put this marker into the cells. And then this was implanted subcutaneously in these mice. This shows this B prime, C, and D. They basically show examples of what happened here. So B is the bright field image of the sponge removed 13 days after implantation. So it's basically they put the sponge in, they wait 13 days, they take it out. And so this is the EGFP expression here. So I guess what that means is it's stable. It's kind of like the sponge acts as like a source of these cells. So that's both stable, the cells are able to maintain themselves in the material, and they can migrate out if they need to. But the point is that those cells are still alive in that material, and you can tell with the markers. So the marker is always the thing we want to see in the image, and so that's just saying that those cells are there and that they're not like invaded by other types of cells. In E, you have this inducer. So this is the sort of the local induction, and then this is the inducer. And so in E, you have a systemic induction of genetic circuits in vivo. Sponges and hydrogels each contain CHO cells stably transfected, were implanted subcutaneously or in the interperitoneum of the arrhythmic or thymic mice. So this is where they put it in the mice again. Then they have this inducer, which is in the drinking water, 24 hours after the surgery, where they put this in. They add this IPTG added in the drinking water. So they're basically using the IPTG to induce these cells. And so that's what they're doing in this example. So, you can imagine like you both want to have some sort of material that you can implant in the organism, have it express or have it be stable, but you may also want to induce it in some cases with a drug that's administered through drinking water or something like that. So you have like a pill or something you would take and induce the expression of this. And so this is just an example. This is where they validate these results. So that's all they're doing here. So they kind of go through and they say that this is a very versatile approach for attaching the genetic inducer to several different classes of biomaterials. This is important because different biomaterials promote distinct morphological and lineage characteristics that have significant application-dependent experimental and therapeutic relevance. By including an inducible circuit, these unique cellular differentiation outcomes can be further enhanced or tightly regulated by modulating the expression of downstream differentiation factors. So you can hook these circuits into other targets within the genome of the cells and get them to express those things as well. So there are a lot of applications that one can use, and they get into this part about using different types of logic gates in these circuits. So it's not just that you can build something that expresses a gene and you can modify that as you want, but you can set up these logic gates. So you can set up an AND gate, which is where you have this logical operation where you take two things and they're activated together. One example is an AND gate circuit in which gene expression would be turned on or off under specific well-defined biological circumstances. The result could be a genetically interactive matrix in which successive cellular differentiation events could be triggered in a spatial and or temporal manner. So you can do a lot of things with respect to sort of differentiation in space and time. And this is, of course, important for a lot of processes that we were interested in. So that's that paper. And then there's this other paper where they're actually looking at what they call temporal transcriptomics. But this is more of a modeling paper where they do this deep dynamical modeling of developmental trajectories. So I think this is a pretty recent paper. This is actually from the bioRxiv. So this is actually where they're developing this labeling method, but they're actually doing this. They're investigating temporal transcriptomics, and so they'll define what that is. But why don't we walk through the abstract? So developmental cell fate decisions are dynamic processes driven by the complex behavior of gene regulatory networks. So we talked about that in the last paper, not exclusively in development, but in this context of these different processes. A challenge in studying these processes using single-cell genomics. So they use a lot of these single-cell genomics technologies where they're able to take a single cell and do look at gene expression, and then, get the sequences of the fragments and map them to the genome so they know kind of where that expression is coming from. But the one of the challenges of this method, there are a lot of single cell methods out there for looking at gene expression, is that the data provides only a static snapshot with no detail of dynamics. So a lot of times the data sets that we get are we can take a single cell, which is a very tiny spatial context, and we can actually get very rich data, very large amount of data. But the problem is it's just one point in time. So a lot of biological processes, unfold over a long period of time. Cell cycle being one of those, where the cell cycle circuit will be regulated in different ways. You have different phases of cell cycle where the cell divides, if you can only capture one part of that process, you're really not getting the full picture. And so that's a problem with a lot of molecular data, as I think we found in our analyses that we've been doing recently, is that it's not it doesn't represent everything going on in the organism. It's just like a very bulk measurement of it. So it's like maybe one point in time, or sometimes you don't even synchronize your sample and you're just getting like a bunch of cells, which is why the single cell context is so useful. You take a bunch of cells and they're maybe in different parts of cell cycle, or they represent kind of a mean behavior of the process. And so it's hard to get that precise sort of information with respect to time. So here we present experimental and computational methods that overcome these limitations to allow dynamical modeling of gene expression from a single cell data. So you need to get the data in place from different points in time in order to get this expression. And you could go, collect the data from different points in time. That's fine, but you have to remember that it's sort of, you have to be able to label these kinds of things that are involved in these processes. So, it's not enough just to get data at different points in time. Oftentimes you have to label things so that they can be identified and normalized. So there's a lot of work that needs to be done to take these data sets and line them up and make them comparable. So this is why this is such a big challenge. So this group developed a platform called SciFate2. So they love these acronyms. And an optimized metabolic labeling method that substantially increases data quality, and profiled approximately 45,000 embryonic stem cells, differentiating into multiple neural tube identities. So they have this system where they can look at cells that start as stem cells, and they're starting to express the markers of neural cells. And this is a very complex process because you have a lot of markers that are potentially useful, but you have to label them so that you can figure out, how they're changing, where they are, what cells they're in, where, what time points that they become relevant, these sorts of things. So they have this labeling technology they're using, and it's able to label different genes, and so they're able to sort of capture this process of going from a stem cell, which has its own sort of transcriptional profile, to some of these neural tube identities, which change their profile in some way. And the question is, we want to figure out which markers or which genes are changing and how. So to recover the dynamics, we developed Velvet, a deep learning framework that extends beyond instantaneous velocity estimation by modeling gene expression dynamics through a neural stochastic differential equation system. So they're using a lot of fancy math, neural stochastic differential equation system within a variational encoder, autoencoder. So they're using a deep learning framework here where they're using, it's sort of this variational autoencoder model, and then they're using these stochastic differential equations within them. So this is basically how they're modeling gene expression dynamics. Basically, they're trying to discover, the different patterns within gene expression by training this model, and then it's basically creating this sort of a dynamical system that's not really like classification, but I don't know exactly. We'd have to look down into the paper and figure out what they're doing exactly. But actually, I think Sushmanth would find this interesting. I know he had to leave, but Velvet outperforms current velocity tools across quantitative benchmarks and predicts trajectory distributions that accurately recapitulate underlying dataset distributions while conserving known biology. So that means that they're able to match what they're measuring with some of the things that they know about this process. So we know these static snapshots of what the genes are supposed to be. Like, we kind of know what the markers are, but we don't know how it gets from point A to point B. So that's what they're trying to capture with this model. That's why we need to use all these deep learning techniques and some of these other things, is to capture that process. Using Velvet to provide a dynamical description of in vivo neural patterning. So they're actually not just looking at the cell fate, but they're looking at this process of neural patterning, because a lot of times, like especially in like the neural tube, differentiation doesn't just happen in a vacuum; it's happening in the tissue. So it's like you're getting these cells; they're becoming neural tube. They're also forming a neural tube, and so in C. elegans, we don't see this as much, but in like mammals, for example, or other vertebrates, we see this where it's in the tissue and it's like forming the tissue. And if you've separated the cells from the tissue, you might not see those patterns of gene expression. Now, that being said, I don't think they account for like geometry. It's just that they're sort of thinking about this process, probably of like the tissue folding and things like that. We highlight a process of sequential decision making and fate-specific patterns of developmental signaling. So they actually have they're working out this temporal process of what's going on in the cell as it's preparing to change its transcriptional profile and signaling different other cells to start to like form this tissue and so forth. So there's this whole process that can happen, and that's what they're trying to model as well. Together, these experimental and computational methods recast single-cell analyses from descriptions of observed data distributions. And so that's where we have these static snapshots of things in time. So you might take a snapshot of a stem cell, a snapshot of a neural precursor cell, a snapshot of another neural precursor cell that's farther along down that set of fates. And so you get that's usually the kind of data we have. But now we can actually move towards models of the dynamics that generate them, describing some of these distributions. So we build a distribution of what these responses should look like to this process, and then model these dynamics using a fancy technique. And then this provides a new framework for investigating developmental gene regulation and cell fate decisions. So that's what they're getting in this paper. So this kind of shows a little bit of what they're doing here. They have SciFate and SciFate 2, so this is like two different versions of this. They label the cells, they fix them, they do all this other stuff, and then they actually do the SCI RNA-seq, which is the single cell method for getting gene expression data. Basically, they chop the genome up into fragments, they measure the number of copies in the cell, and then they map it to a draft genome so they can figure out where it came from, what the transcript is. And then they do these dimensionality reduction plots, just a low dimensional visualization. I guess that's a PCA. So it's just a normal PCA that shows basically these in different categories. So you can take all the different transcripts from different 44,713 cells, and you can put them in get these in these categories from these principal components. So you have mesoderm here, early neural cells, and MP cells, and so forth. So this shows that like their method is sort of validating their method, that they're able to measure what they say they're able to measure, that their markers are consistent with like different cell types, and they cluster together. So the idea would be like, if I think it's a neural cell, it's not enough just to say it's a neural cell. I need to show that it's a neural cell, that the markers, other cells with the same markers cluster in the same places, and then I can compare it with other types of fates, other types of gene expression profiles, and you get a different, you get these different clusters that come out of it. So that's what they're doing there. And then they're, they're training this model. This is what they call VELVET, which is deep generative velocity inference. So they have this, so to model gene expression dynamics, we focus on a generalized idea of velocity that is not specific to splicing dynamics. So they apply a biophysical model of RNA transcription, labeling, and degradation. We define a cell's velocity in terms of three observables: labeled reads, total reads, and labeling time. And these are technical details, but and then they have a free parameter, and then they have something, this represents the gene-specific degradation rate. So they're able to look at the velocity of like a RNA transcription. They're looking at like the number of transcripts and then sort of their degradation, and that should tell them something about like, how long a transcript will be around. If it degrades and then there's another gene that gets expressed associated with another profile, if those get expressed, those copies will be around for a certain amount of time and then they'll degrade. And the idea basically is that you have these pools of RNA. They get produced when a cell is in a certain state. So a certain marker, say for a stem cell marker, it'll be expressed at a certain amount. It'll produce a certain number of copies of the RNA. And then those copies will degrade over time. But if the cell is still a stem cell, then it will retain the sort of stem cell fate, and it'll like keep producing copies. So even though it degrades, you still get copies and you still have that upregulation of the stem cell marker. But on the other hand, when the stem cell transitions into a neural cell, some of these stem cell fate genes stop producing copies of the RNA. So then that RNA will be around, but only for a while until it degrades. Then once it degrades, it just goes back to a baseline level, and the neural markers will start to ramp up with respect to copy number, and those will maintain their rate of renewal. So, the number of copies will degrade, but then you'll still have copies left over. So that's the idea. They're looking at this velocity of the marker, velocity of the RNA copies, to see, how long it sticks around, and eventually as the cell state changes, it'll the profile will change, but it's not instantaneous like we might think from something that's the static measurements. So it doesn't just switch very abruptly. You have these dynamics where you get things that are upregulated and then they kind of fall in number, they decay, and then there's another marker that comes up, and then there are these interactions that happen as one marker is shutting off and another marker is turning on. That can be interesting, but that's what they're trying to model. So to get a little bit more context about the temporal transcriptomics paper, I'd like to do a little bit of drawing on the board to show what these kind of experiments look like over time. So a lot of times in transcriptomics data collection, you have, in this case with next-gen sequencing, you have your sequence, which is of course the DNA, and then you have RNA being expressed in different locations, different transcripts that are collected. You get these fragments of transcripts, and then you get a summary, usually, of transcripts at different locations. So this is our copy number. And we'll just say that, we have 3 examples here where the copy number is variable over time, or actually just in one snapshot of time. So this is the gene expression is the number of copies that you get for a certain fragment or a certain site. And so then you can look at those over time and these sort of static summaries that we usually get where you sample cells at different times. And for these different locations, you get different changes in how they're upregulated. So that's usually the typical sort of snapshot that we get. We might look at this point versus this point and say that there are many fewer transcripts at this point versus this point. What they were doing in the paper we're talking about today was they were looking at the copy number, but they're also looking at it over time dynamically. So if this is continuous time here on this axis, then we have, this is the copy number at this time point here. And this copy number is dependent on sort of the production of, so if we look at a dynamical representation here, and we have our time axis and we have our copy number on the left, this copy number gets produced at a certain point in time, and that reflects the activity of that the promoter on that stretch of DNA producing RNA. But what happens if you stop producing this? So for example, if you have a stem cell that transitions into a neural precursor, that stem cell marker shuts down its production of RNA, and so this marker then begins to decay. The copy number falls over time as it stops being produced, or at least at the level that it was originally producing at. So you get these time points where you get this reduction in the amount of transcript, and they call these RNA pools. And there are experiments you can do where you actually either, change the fate of the cell, or sometimes there are experiments where you can shut down the transcriptional machinery in the cell, and then all transcription stops. So you can watch the decay profile of a certain gene. So you can measure the transcript for that gene in terms of the RNA. You can look at it as the cell's decaying. So you shut off the mechanism and then you sample after a certain amount of time. Usually it takes 48 to 72 hours for decay to be complete. So after that time, you end up with a negligible amount of the transcript. And so that basically happens in this process. So you end up with a very low amount after that time, if it stops producing. But of course, for most cases, your marker will continue to produce RNA. Some of it will naturally decay because there are copies that are older, but there are also copies that are younger, so they'll replace them in this pool. So this pool gets replenished. And so this time point here, this will remain stable over time. So we can actually redo this figure where we have continuous time here. We have this transcript being produced at this copy number here. And there might be fluctuation around that copy number over time. And that reflects the cell staying in the stem cell state being maintained there, but also, having a replenishment of transcripts relative to the natural decay of these transcripts. So this profile is much less decay-like. And so if we actually think about like what happens when like one marker shuts off and another turns on, we can represent one in circles and one in lines. So this is the stem cell marker. Say it gets shut off because there's a transition to a neural fate. This starts to decay because it's shut off. Then the neural marker starts to ramp up production. And it might do so unevenly for a while, but then it starts to ramp up. And the same dynamics are the neural markers subject to the same dynamics as what we saw in the last example, where you have production of the transcript, but you also have decay, natural decay, and eventually you end up with this profile. So you transition from one marker to another marker. You have this transition of state. And that's how they're measuring that. So there are other ways you can measure the transition of state. You can look at the morphology of the cell, but this is the transcriptomic profile. So that's like the state of the art in thinking about that problem in terms of transcriptomics. You get this temporal sequence that is, something we can study, and so it's maybe a different view of the world than just looking at these static measurements. Okay. Well, that's all I have for today. Any comments or questions?
I met someone online one day. It was a conference. Anyway, he was looking at the brain and looking at cells in the brain like that. He was plucking individual cells out and checking their genomics.
Oh, yeah.
I thought that was very tedious, that he takes slices of the brain and freeze dry them or something, or freeze them, and then pluck the genetics out.
Oh, yeah. Yeah. These genomic technologies are getting more and more affordable, I guess you could say. So it's like you can do all sorts of really interesting stuff now that you couldn't, like, maybe even 5 years ago.
Oh, a lot of research. I wonder where it's going.
Yeah. Well, some people criticize it and say that, like, those maps that I showed you of the PCA and of their other types of maps you can build, like UMAP and other things, that they criticize them for being kind of like, can you distinguish these things from random patterns? Because we kind of think, well, we throw these data into a dimensionality reduction algorithm, and get a pretty picture, and it tells us basically what these transcripts mean, or what this data means. And there are some people who think that we don't really know what they mean. So we can have a lot of data like that looks we think it's very precise, but sometimes, it may not be as precise as we think it is. Yeah.
Wow.
Yeah. So it's like something to keep in mind with a lot of the data analysis that you might see, that it's really, our methods are really early in the sort of scope of things, especially when the technology moves really fast. You say, well, we can use this method. This is a good method for doing this. And of course, you want to really have multiple methods for analyzing these, and maybe some tried and true ones, but there isn't really anything like that yet. So,
Well, you need several different methods, and they all have to converge. So.
Yeah, it just takes time. Yeah.
Okay. I'm giving a presentation today.
Oh, good luck.
To my advisors, and hopefully they like it. I don't know.
Yeah. Yeah. Well, good luck. Yeah. And this is on the salt integrity stuff, or on?
Yeah, it's tensegrity project, my candidacy project. Well, it's just a progress report.
Okay.
And I should be done the tensegrity project by now, but I've had interference.
Yeah.
So.
Okay, thanks a lot.
Thanks. Talk to you next week.
Bye.
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 Bye. Okay, now I'd like to talk about a few papers in an area I'm calling symmetry and inference. And so this has to do with inferring mechanical properties of the cells and looking at the cell symmetry and looking at this in embryos and in tissues. So this first paper is from Development in 2021. This title is Inferring Cell Junction Tension and Pressure from Cell Geometry. So this is inference by the experimenter. And they're trying to figure out how to measure these variables. And they're getting this information from cell geometry. So the abstract reads, recognizing the crucial role of mechanical regulation in forces in tissue development and homeostasis has stirred a demand for in situ measurement of forces and stresses. So there are two contexts here. There's tissue development, where your cells are coming together and they're changing their shape and they're migrating around. And then homeostasis, where the tissues have to maintain their integrity. And there are different ways that this happens. They're distinct from development, but they both require this in situ assessment of measurements of force and stress. So among emerging techniques, the use of cell geometry to infer cell junction tensions, cell pressures, and tissue stresses has gained popularity owing to the development of computational analyses. So they're doing these computational analyses from the simple measurements, and they're getting basically they're able to do these kinds of higher level inferential activities. This approach is non-destructive and fast and statistically validated based on comparisons with other techniques. However, its qualitative and quantitative limitations, in theory as well as in practice, should be examined with care. This is a primer that they've designed for people in developmental biology to summarize the underlying principles and assumptions behind stress inference, discussing its validity criteria, and providing guidance to help beginners make the appropriate choice of its variants. We did extend our discussion from two-dimensional stress inference to three-dimensional. So we talked about three-dimensional tissue engineering earlier in the meeting. This is where we go from the two-dimensional case, which is like a culture dish, to three-dimensional case, which is like the actual tissue. And so these are two very different environments. Use the early mouse embryo as an example and list a few possible extensions. So this is an attempt to make this more accessible to the community. And again, this is mostly for developmental biologists. I don't know if engineers, I guess, are maybe more receptive to the idea of computational modeling in general, but this is one of these things about the audience, as I said before. They're asking different questions. They're trying to understand things for different ends, and so it kind of means that you have to. Sometimes you don't run into certain methods that other fields are sort of come as second nature to another field. So the role of mechanical interactions during morphogenesis goes way back to the turn of the last century. Darcy Thompson, of course, mentions it in his work on the mathematics of phenotype. And so forces are generated by molecular motors, notably by actin-myosin networks, and are transmitted via cytoskeletal elements through cell-cell adhesive complexes. So we've talked about these networks before. They're within the cell, they're in the cell membrane. They're these actin-myosin networks, which act similar to muscle in a lot of ways. They're flexible and they produce forces, and they have to interact with other cells, so the cell can change its shape, but it can also generate forces for migration or for sort of maintaining its own space, whatever it's used for. There are these adhesive complexes that mediate the connections between cells, and those are important for a number of reasons. Stresses are the effects on these internal forces, measured at the cell or tissue scale. So when you have a tissue, It's a bunch of cells that are adhering to one another. The adherence of the cells is important for the integrity of the tissue. So we want to measure the different things that are affecting those connections and affecting the entire collective structure. So the first one is stresses, and these are the effects of internal forces, so the actin myosin elements pushing against one another. And this is critical to things like tensegrity because you generate these dynamic situations where you have dynamic tension. Stresses are part of that. Forces and stresses contribute to the determination of static cell shapes and packing, as well as dynamic changes in cell size, shape, number, and position, and gene expression. So we have all these other changes. We have cell size, so as this tissue grows in size, the forces change and everything else. You have shape, of course, if it's packed in a certain way. It's a sphere versus a sheet. Number and position, and gene expression. Of course, gene expression is something we've talked about as affecting sort of the state of the cell, but sometimes gene expression can be moderated in different cells by external stimuli, like the chemical microenvironment or something like that. And so all these ultimately contribute to tissue morphogenesis. So this is why we want to understand mechanics. And there are a lot of ways you can measure this. You can use things like contact manipulation, which is things like parallel plate compression, atomic force microscopy, and micropipettes, basically making contact with the cell and manipulating it that way. Manipulating using light, so you can use optical tweezers or laser ablation. You can use visual sensors such as FRET sensors, which use proteins to sort of as a target liquid droplets and elastic beads, and non-mechanical observation techniques. So these are just ways you can do imaging that allow you to observe what you want to observe. And so these are all ways you can measure these stresses and forces. And so some of these are invasive, some of these are less invasive, so you have to decide based on your experiment what you want to know, which one to use. So it's really always a matter of choosing the right tool for the right job. Stressor inference is a very promising approach to link mechanics and developmental processes because in stress inference you need to make the minimal measurements. Sometimes they're non-invasive, sometimes they're non-interfering with the system, and then you just get the minimal measurements. You plug them into the inferential engine, which is usually a simulation or some sort of computational technique, and you can model the result. So that's basically the idea here. So it's helpful. It's helpful for biologists because it doesn't interfere with the system. It interferes minimally, and especially if it's in vitro, or in vivo. It can be hard to actually get in and actually do manipulations. So this is always a good way to go. So there's this glossary of different terms. They actually use Bayesian methods. They mention Bayesian methods here. It's interesting. And so they actually talk about using stress inference to study development. So this goes back to Darcy Thompson in 1917, where cell stresses can be inferred from images of cells within epithelial tissues. This was implemented, though, more formally the first time by Stein and Gordon in 1982. So this is a froth made of soap bubbles. This is a well-known example of a pattern with equal tensions. So you have this froth of soap bubbles. It forms a pattern. The pattern has equal tensions. Analyzing a 5 bubbles cluster, Stein and Gordon checked the tension values they inferred were equal within their position, hence validating their approach. Using this approach, they further analyzed a patch of 11 cells in the superficial cell layer of a frog gastrula and revealed significant differences among cell junction tensions. So this is where they have sort of the soap bubble model, where they have equal tensions that form a pattern. They were able to infer that these were equal. So they were able to validate that approach using the soap bubbles, and then they went to a model of the frog, the superficial cell layer of a frog gastrula, and they were able to measure or see that there were significant differences among cell junction tensions. So it's this example using a sort of a control system or a sort of a at-rest system or random system or whatever you want to call it, and then using something that has some structure that's biological as sort of like the condition where you're doing the intervention. So it's like you have a system that isn't biological and one that is. You know the one that's not biological is uniform. Now what's the effect of the biology or the structure in the biology? In the 2000s, with progress in computers, live imaging and finite element modeling, Brodland and co-workers revisited this principle to infer tensions from cell-shape images. We've talked about Brodland in the meeting in a number of different cases. In cases where limited information was sufficient to provide insights into the underlying cellular mechanisms, angles between cell junctions, or roughness of clone boundaries were used as indicators of ratios of junction tensions, while cell junction curvatures were used as markers for cell pressure differences. So they're able to use these different features of the image to infer these differences and some of the ratios that you might want to know, like if you want to look at different parts of the image and see that those visual features change, you can infer that those are due to these different effects of tensions and forces and pressures. So this is something that, you know, we can basically take from an image, so we can actually infer stress, because we know that the laws of physics are, you know, pretty predictive. We can just use equations to model some of these changes and assume that the material changes in a certain way when exposed to these sorts of physical parameters. And so this is stress inference, basically is represented by its current variants discussed in the primer and were first introduced by Chiao in 2012. It's been extensively used in 2 dimensions to obtain mechanical information, mostly on planar epithelial tissues, meaning that it's this flat surface of epithelial tissues. So, you know, really you need to have like a benchmark for this. Works best when you have a way to say, this is what the profile should look like in a control condition where it's very simple, and then we can make these inferences, and they should fit the observed values should match the predicted values. That's how you do the analysis. In particular, it has provided important insights in the mechanics of Drosophila egg chamber formation, pupil dorsal thorax, and wing morphogenesis. So this is all in Drosophila, different aspects of its development. And so this is, they're able to match this with parts of Drosophila development, as well as embryo ventrofurl formation, germ band extension, and germ band retraction. Stress inference has also been applied to study avian development, revealing tissue scale pressure gradients in the quill primitive ectoderm, and mechanical heterogeneity associated with hair cell differentiation during avian cochlea formation. So this is, again, this idea that in biology, these mechanics are heterogeneous. They're not homogeneous like in a soap film or something else. There's actually the structure that's associated with these processes. So this is the mechanical heterogeneity here with hair cell differentiation. And there are many examples where you can actually take images and like basically infer what the physical parameters should be. So they talk about a number of cases here. These are some nice images here where they show examples of stress inference and development. So A is junction tension distribution in the Drosophila egg chamber. So you can see this where you have this heterogeneity in the image, and you can infer forces from that. B is actually a map of tissue stress for the Drosophila pupil dorsal thorax. So this is a tissue. We've seen these kind of maps before where you have this parameter that has a value and it's going in a certain direction. So it's like this force map where the forces are applied in different directions. And you can see this is where you get this sort of, you get the furrow here, and you get these different changes in the direction of forces. And you can tell from looking at the image what direction the forces are being applied, because sort of the sort of the layout and shape of the cells underlying this. So that can all be inferred, and you can build a map like this and show that there are these differences throughout the tissue. C and D are overlays of color-coded inferred junction tensions. So C is an inferred stress for each plotted cell as an ellipse, and D is on a confocal image of Drosophila lateral ectoderm near the cephalic furrow during late-term band extension. So these are the two examples. These are the stresses here, these circles. These are the inferred tensions as a heat map. So you can see that the tensions are higher on the outside and lower on this inside area. And again, if you look at the images, you can tell the shape of the cells and the orientation as to what the tensions are. It just translates right into the image. And then E is a map of inferred pressures of the primitive ectodermal quail embryo. So this is where you have this map of, this heat map that gives you the values, and then this is the quail embryo, and this is sort of the distribution across space. So this is, this just shows kind of these different applications of these methods that they're talking about in the paper. And they have this thing called dynamic stress inference. The central hypothesis underlying stress inference is that cell shapes result from the balance of cell-cell interactions through junctions between two cells and at vertices where the junctions meet. So you can see these cells are all sort of these geometric shapes, and they all have vertices, kind of like, you have a corner, say like right here, and you have like three cells meeting at that corner. And that corner is a vertex of a network. It's a mechanical network where forces and stresses are transmitted across them because it's just kind of the edge of the cell, and cells are meeting at that edge, and so the edges come together in that vertex, and then the shape of that, of course, is varied by the amount of forces and stresses that are experienced there. So you can see there's a lot of heterogeneity in terms of the sort of where these vertices are with respect to the edges. The cell shape changes, and you can see in the color coding that the cell shape changes as this value changes as well. So, there are a lot of ways you can infer it from just looking at the image. It's kind of like when you can look down on an area and look at the geologic, you can kind of infer the geologic activity from aerial images of a landscape. You can look at like different features in mountain ranges or in valleys, and you can see different things that may have happened in the past. And that's sort of the idea here. Except this is very much in the present. So this is, basically the method. So again, you don't just infer things from the images, but this is the idea behind some of these easier sort of, taking things and running it through a computational model and getting the answer. You don't have to do a lot of direct manipulation with this. So this is an example here of some applications of stress inference in living tissues. There's this paper, Shu in 2018, where they talk about C. elegans early development. And this is about tension and pressure inference, sensitivity test and inference scheme. So they actually do this in C. elegans. They've done this in zebrafish, mouse, avian cochlea, quail, primitive ectoderm, and of course in Drosophila. Stress inference can't really give us the answers to everything. We can gain information from stress inference, but it's not all-encompassing. It doesn't tell us everything we need to know. So there's some images that sometimes deviate from mechanical equilibrium. So one of the main ideas here is that there's this mechanical equilibrium. And when the tension or stresses deviate from that, you can see the effects and the boundaries between the cells or the orientation of the cells. Now, there are different cases where maybe that isn't informative, or maybe it's just not, that useful. It's hard to really kind of make an inference from that. So here's some images here that are sort of that they've identified as maybe not representative of where we want to go. So there are these crenellated edges. There are these S-shaped edges. You can see the S there. You have edges with a constant sign of curvature, but not a constant value. So it's not a circular arc, it's kind of a compound circular arc. D is plant tissue with a very irregular cell shape. So plant tissues are usually like square or sort of this elongated rectangle, but sometimes you have these irregular shapes in plant tissue. So you don't really know, the junction is wiggly and you can't really infer much about the forces from that. You have the triskelion, which is the curvature of the three junctions that meet having the same sign. So this is where they're all sort of, I guess, positive. And so there are a number of things you can look at the junctions, and sometimes they're very easy to read and sometimes they're ambiguous. So that's what they're getting at here. And this is actually where they talk about signs. So these are all like different, the way these are sort of abstracted from the image. So when you have a junction, you have these sort of edges that radiate from them. The edges then have different forces. They can have, or pressures, they can have a positive or negative sign, and so, or an angle even. And so then, we can measure these, but, sometimes these are not, like sometimes they're all the same sign, sometimes they're negative. So the idea being that, like, sometimes they violate the physics. The second paper is actually more a research paper, not a tutorial. This is continuous symmetry measure versus Voronoi entropy of droplet clusters. So we've talked about droplet clusters, especially with respect to early life, and also with respect to archaebacteria, in like inferring some of the shapes of these organisms. But this is actually doing these on droplet clusters. And so this kind of talks about sort of the physical chemistry of this. So the abstract reads, symmetry and orderliness of two-dimensional levitating microdroplet clusters are quantified with the Voronoi entropy. So there are these Voronoi packings, and you can see that they have these sort of hexagons that are packed together, and they come from a centroid. So the centroid, you go out from the centroid, and the edge is like the midway point between the centroids, and then the packing emerges from that. So you end up with these hexagons, which is the most efficient packing around each centroid, the centroid being the droplet. So then, there's this Voronoi entropy measure, which we'll find out what that is. That's what they're quantifying this with. And then the continuous symmetry measure, which is CSM. So they're measuring symmetry, and they're measuring this entropy. The time evolution of both V and SCM is investigated. To compare the correlation between the two measures of orderliness, the Pearson correlation coefficient is used. The maximum correlation between V and CSM was found for clusters containing the number of droplets, enabling the formation of hexagons. So droplet clusters possessing six-fold symmetry. So what they're doing here is they're looking at these droplets. They're sort of making ripples against one another. They kind of intersect somewhere. And they're using entropy and this continuous symmetry. So they're saying that symmetry is important. This distance between droplets is important. And with these measures, they find that hexagons are the best fit. So this is actually six-fold symmetry. So you have these six-sided hexagons. There's the six-fold symmetry in the hexagon. And so that's the best fit for this, for what we're looking at. In other cases, the maxima and minima of the V and CSM are not only well correlated. Moreover, in certain cases, maxima of the CSM may correspond to the minima of the Voronoi entropy. Symmetry and orderliness of 2D patterns could not be quantified with a single mathematical measure. So this is what they were trying to get at. They were trying to figure out, these clusters, how to characterize them. And they found that using Pearson correlation coefficient, which isn't the perfect tool for this, but that these are maximally correlated. But sometimes they're not. And so, in some cases, this is not the best fit, but this is basically what this should look like. And so they're really analyzing patterns here. So it's usually implied that symmetry changes abruptly or intermittently. In other words, symmetry is viewed as a binary feature when an object is considered either symmetric or asymmetric. So there's this idea of symmetry in developmental biology as well, and it's basically where things have pairs and they're well ordered or whatever. And then there's this asymmetry, which is where the symmetry is broken, and there's this, things are not even between like the different folds of symmetry. So you can have, like in a bilaterally symmetrical organism, you can have two sides that mirror one another. And then when that symmetry is broken, you have an asymmetry that maybe leads to a change that is, differentiated tissue or something. But a lot of times symmetry is this sort of subjective binary feature. You need to have a mathematical method or technique to understand, what that looks like. And so what they've done is they've done this with these droplets. They've put the droplets down. They're fitting it with a model of symmetry, and they're able to get this sort of this order. So what they're using here is this black-white paradigm. This is the continuous symmetry measure that they're interested in. Such an approach to symmetry broke the black-white paradigm usually adopted for this analysis of symmetry of patterns. So this is actually breaking the traditional model. We implement this approach for the analysis of symmetry of 2D droplet clusters levitating above heated water vapor interfaces. So they're able to look at this. Large clusters form hexagonally ordered honeycomb structures similar to colloidal crystals, while small, from 1 to several dozens of droplets, clusters possess special symmetry properties. Small clusters, in particular, may demonstrate 4-fold, 5-fold, and 7-fold symmetry, which is absent from large clusters and crystals. The symmetry properties of small cluster configurations are universal. That is, they do not depend on the size of the droplets and the details of the interactions between them. So this is interesting. This is not only this sort of packing, but they also have unique properties that they found with the system. And so this is a typical droplet cluster levitating above the water surface. You can see that it looks like just kind of a sphere with a bunch of droplets sort of distributed throughout. But you want to use a Voronoi tessellation to find sort of the boundaries between them, to find the packing. The Voronoi tessellation, or the diagram of a 2D area with a set of nodes in it, divides the area into a polygonal zone of cells, with each cell containing points that are closest to a given node. Such tessellation represents the two-dimensional special case of the Wigner-Scheitz cell tessellation. They cite that here. We calculate the CSM of a Voronoi diagram of a droplet cluster, not the CSM of the cluster itself, considering the theorem stating that a Voronoi cell always has the same point symmetry group as the underlying lattice of the two-dimensional pattern. So they define the continuous symmetry measure here, which is a function of the minimal average square displacement of the points m sub I of the shape, those that have to be undergone to acquire the prescribed G symmetry. So basically these droplets have to be displaced, all of them at the same time, across the shape they have to undergo these kind of transformations to acquire a certain symmetry. So it's basically where you want to, they have to organize themselves so that the symmetry can be achieved. What is the displacement? And so this is the calculation of the CSM for these triangles here, and then this is the centroid here. So this is the common centroid point O. And so then this is an example of the Voronoi diagram corresponding to the droplet cluster. It's basically where you find the sort of the midpoint between the any two droplets. So when you have any two droplets with an interface, it forms a boundary. So there's an interface between this droplet and this droplet, this droplet and this droplet, this droplet and this droplet, and so forth. And then these two boundaries kind of meet. We can actually see where these two Voronoi spaces meet. So this is what they call Voronoi packing, where you get these distances, the midpoint between the two any two clusters, when you have a bunch of clusters together, you form these shapes. And so they found that the hexagon was the best fit for this. So these are a couple of examples for these different packings. And then they show here the Voronoi entropy versus CSM times 10. The red curve corresponds to the time dependence of the CSM. The blue curve corresponds to the time evolution of Voronoi entropy. And so this is how they define Voronoi entropy. It's the fraction of P sub I is the fraction of polygons possessing n edges for a given Voronoi diagram, also called the coordination number of the polygon. And I is the total number of polygon types with different numbers of edges. And then so they try this for 10 droplets, 19 droplets, 34 droplets, and 51 droplets. And as you can see, you start to get this sort of less fluctuation over a larger number of droplets. There's sort of maybe a little bit more correspondence between the blue and the red functions. And the blue function doesn't drop to 0 at any point in these two. So the more droplets you have, I guess the more stable these measures are. And so this is an example of Voronoi tessellation built of only of hexagons. So you can see at the end you have these Voronoi tessellations that emerge. You can measure them and characterize them with these two measures. So that's all I have for you today. Hope you learned something.